first we start here in the nucleus. Now this is a chromosome. This holds the genetic information for genes that will eventually be expressed as phenotypes. This right here is the gene itself on the chromosome. This is all held in the strings, which is the DNA, or deoxyribonucleic acid. Now over here we have a model with these white strings as the DNA and its double helix formation. This blue string is the RNA's transcript, and that's coming off of this RNA polymerase protein. The polymerase attaches to the DNA and it slides along it, undoing the double helix, creating this, and then redoing it. So I'm going to show you that right now. See, it slides along the DNA, creating this RNA transcript. Now, when DNA transforms into RNA, it's called transcription. And some differences are RNA has ribose as its sugar, where DNA has deoxyribose, and RNA has uracil, a U nucleotide, while DNA has thymine, a T nucleotide. Now this RNA transcript isn't quite ready to go out of the nucleus, so it's going to come down and go through some RNA processing. In this processing, it loses its introns, and the exons stay connected together. And also, at the 3' end, it gets a poly A end, and on the 5' end, it gets a 5' cap. So now it's ready to leave the nucleus. It will slide through this nuclear pore and into the cytoplasm. Now down here I have another model of the mRNAs. Okay? So here I've added on all the nucleotides with the information that it's going to express. This first one, AUG, makes methionine, or the start codon for the sequence. And then there's some others to carry this certain amino acids. And this last one, UGA, is a stop codon. Okay, so this is going to go through the cytoplasm until it comes over here, lines up on this ribosome, and starts the next process called translation.
Okay, so with this chain, we're going to start off with the process of translation with these two ribosome subunits, the larger one up top and then the smaller one on bottom. And so we have the start codon, which will attach to an anticodon right here of tRNA. Now this tRNA, where did it come from? Let's move over here, and it comes from an amino acyl tRNA synthetase. And this is where the tRNA attaches to the starting amino acid and will gradually move over to this RNA subunit to start translation. Now this one connects to the A site, which will be the initiation, where it comes down and the anticodon codes for the start codon right here, creating the first amino acid. Then elongation occurs with the P site right here, and we gradually move along the chain to where it hits the stop codon right here. And this will be at the E site, where the final stop codon releases this tRNA. Now as this is going on, the polypeptide chain is growing more and more until it hits the stop codon. Polypeptide chain elongates and finishing off protein synthesis with peptidyltransferase making these covalent bonds in between the polypeptide chain, allowing it and completing protein synthesis.